So first of all, I would like to thank everybody for being here. I mean, I've created with Alois and Henri Ceres Brain Therapeutics 4 years ago now, and this is a yearly rendezvous that we all love. And so thanks to Carol, Frank, and the ACD who are present, I think it's very inspiring for everybody in the room. So before going there, I use the analogy of the steeplechase race for developing a drug in CTD. We are riding our horse with Ceres. There are a lot of obstacles, a lot of ditches, a lot of fences to go across, but we are still on our horse and we are still riding. So it's a long journey, but we are going toward the IMPD IND. It's not yet there. We hopefully will not fall from the horse, and we are still going. I don't know if anybody knows the steeplechase race horses. It's coming from Ireland, and actually it was a race between small villages, and the horse, the competitors on their horse were going from one steeple from a church to another steeple from a church. So this is why it is called steeplechase horse racing. I will not present any more Ceres. It's a very small company, but still we are a lot of people now. On the right-hand side of the slide, there are the permanent people in the company: Henri, myself, Alois, Clémence, who is here, and Clara. And then we are working with experts and consultants. We dedicate a lot of time for the development of the drug. And we have been recently joined by 2 members of the board. One guy with Charles is a Sanofi guy, one of the top leaders at Sanofi, and Marie, who is one of the top leaders at BCG. So the key question is how to bring creatine into the brain neurons. We have been talking about that for all the 2 last days. Our approach is to say that there is 2 barriers that are really almost physical barriers, very difficult to cross. The first one is the blood-brain barrier between the blood vessel and the brain. But there is a second one that we don't need to neglect. This is the barrier at the level of the neurons. And the key thing is to be able to bring the creatine within the neurons. It's not within the brain, it is within the neurons. And this is very important. So our approach, and I presented this approach, Alois presented this approach already a long time ago, is to put our product in the nose, in the nasal cavity, where it will enter the olfactory nerves as well as the trigeminal nerves. From there, already in the nerves, it can reach the origin of the neurons, so the olfactory bulb as well as the trigeminal core, and then diffuse from one neuron to the other to the whole brain. And this is something that we've already proved. How do we do that? We do that by bringing different technological bricks, innovative technological bricks together. The first one is that we have a specific creatine ester synthesis that allows us to have a specific creatine ester. It's a dodecyl creatine ester. However, the way it is produced, which is patented, gives us the opportunity to prepare a specific formulation, which is a particulate emission, and this particulate emission can be sprayable in the nasal cavity. Then it is the application of the nose-to-brain pathway. The nose-to-brain pathway has been described during the 90s. However, it has never been applied to a drug so far, and Ceres Brain Therapeutics will be, hopefully, the first company to claim it. And so there is a regulatory pathway to build with the regulatory authorities. So take time, but we will arrive. What did we show? First of all, we showed that we were able to bring creatine to the brain. But if you remember a few minutes ago, I said the brain is not enough. So we showed also in monkey that it was not only in the brain, but in the neurons in the brain. And this is very important. And we found our drug, CBT-101, as well as the metabolite of our drug, creatine, in all the different areas of the brain. So we demonstrated that we were able to bring creatine into the targeted cells, the brain neurons, meaning that we don't need actually a lot of creatine in the brain. We need a bit of creatine to go in the targeted cells. And this cannot be showed on spectrometry MRI. Why? Because the quantity of creatine that we bring, which is useful because it's in the targeted cells, it's not a lot of creatine. It's just what you need. The creatine within the neurons has different effects, and this is what is important, is that it works. So what we showed is that the brain plasticity was increased in cow mice from Laura Baroncelli as well as Matthew Skelton, so 2 different models. We were able to show that there was an increased expression of synaptic biomarker. In the cow mice of Matthew Skelton, we showed, and Nicolas Tournier presented the result yesterday, that the cerebral energy consumption was preserved while the mice was aging. And we also showed in the different models that the cognition through the novel object recognition test was improved. We also used some other models, which were not SLC-A8 models, to show that the creatine within the neurons has an impact on the brain. And we used a model for Parkinson's disease, 6-OHDA model. In this model, you create a dopamine infarction in an hemibrain of the mice. And if you treat your mice before doing the infarction, actually, you preserve the brain and the dopamine cells within the hemibrain. And so the 6-OHDA treated mice were preserved of the infarct. The dopamine production was increased compared to control. So there's really an action of our creatine within the neurons. In parallel to this dopamine production preservation, the behavioral test showed that our mice did have a lower Parkinson's syndrome compared to the control. Again, parallel between brain plasticity result, biology result, and clinical result. So we do think that today we have a product that is able to deliver creatine within the brain cells, the targeted brain cells, and this product has an effect, a biological effect, a metabolism effect, as well as a cognition and motor function effect. Our approach is very simple. You can drive your car forever if you stop at each gas station and you take a few liters. You see, everybody can experience that. It takes time. You just have to stop, put a little bit of fuel in your engine, and then you can go for 20, 50 kilometers, stop at another gas station. And we think it is the same. CTD neurons lack creatine. We propose a treatment where you give to your child, to your patient, or son, even daughter, why not, the drug. The drug goes in the nasal cavity, from the nasal cavity in the brain neurons, and then it can be used by the neurons. At some point, the kids will be missing the drug. You can give back the drug, and so it will be a chronic treatment for life, probably. However, we will support the brain neurons with the missing creatine. The way it will work will be very simple. It's a nasal spray. So here's a nasal spray. And here it is. You put it in the nose like that, upsheet in both nostrils, and it's done. Whether we need to do it every morning, and noon, every morning, noon, and evening, we don't know yet. And this is things we are working on with Henri and Alois. So where do we go from there? We are working on the CMC, and we are working on the toxicological study. The CMC, the scaling up of the formulation, has been achieved. So we are very proud of that. We have a formulation, very specific, patented, that is stable. We are working on the short-term clinical study to be able to conduct a phase 1 study in the coming months, hopefully not years. I will not put a date really on that. And the key thing is, as Melanie said, it takes time. You know, this is still a steeplechase horse racing. We see the church steeple. However, we don't know how many fences. We don't know how many ditches. We don't know how many obstacles we need to go across. But hopefully, we will have good news in the coming months. And then regulatory authorities will have to review our dossier in Europe, in the US. We propose a phase 1 in healthy volunteer. We have already designed the phase 1. It will be single ascending dose followed by multiple ascending dose, quite classical in drug development. And then we will go for phase 2 in patient to assess efficacy in CTD patient. Melanie just before me, but during the World Conference, many questions were raised about this clinical study design. So we do think at Ceres that the SCED is probably a good way to avoid the bias of the heterogeneity of the patient. We need to continue to talk all together as a community to design the best phase 2, to be sure that the regulatory agency, and Andreas Schluss was right on that. The key thing is to design a study that allows to demonstrate an efficacy and that the regulatory agency recognizes as a success. Because without the regulatory agency recognizing it as a success, there will be no drug for everybody. There will be only drugs for the people participating in the study. So this is key that as a community we work together towards this goal to hopefully have a drug for your kids, sons, girls, in the next years. So thank you very much for your attention.